Okay, we want to pick up with Chapter 4, talking about enzymes and metabolism. And when we do, these are our learning objectives. We're, of course, going to define metabolism and anabolism and catabolism. We're going to talk about the structure of enzymes and their function and what we mean when we say that they are catalysts. We're going to talk about activation energy and how the substrate binds to the enzyme, and how enzymes can become denatured and stop working. So this leads us to vocab metabolism. This is defined as all of the chemical reactions that occur in your body. And the chemical reactions that happen depend on the activity of the enzymes. The amount of fat that a person stores depends on how fast or how slow they break down their food molecules. Enzymes regulate all of our metabolic reactions. They are proteins, we learned this in Chapter 2, that catalyze or speed up metabolic reactions. And they're named for the reaction they catalyze. And they'll end in the suffix ase a lot of times. Sucrase is an enzyme that breaks down sucrose. So when they are catalyzing these reactions, they may be breaking down molecules or building them up. This is how we break down our foods and ingest and get the energy from our foods, from the chemical bonds that are there. Now, activation energy is defined as the energy required to start a metabolic reaction. And what enzymes do is they decrease how much activation energy is needed to start it. That activation energy is a barrier, and that keeps all chemical reactions from happening constantly, even when we don't need them to. Enzymes don't need heat to get reactions started. They break down break the chemical bonds without damaging or killing the cells. So the reactions happen more quickly. If you look here at this picture in Figure A, no enzyme is present. So to get over that hill, it's a pretty significant size hill. It's going to require a lot of energy without the enzyme. But in Figure B at the bottom, with the enzyme present, look how the height of the hill is reduced. It doesn't take nearly as much energy to get over that little bump as it does that great big hill up there in Figure A. But we need to remember that enzymes are very specific. They only work on specific chemical reactions, and it has to be the correct enzyme for it to reduce the activation energy. Because without activation energy, these metabolic reactions be happening all the time, even when we don't need them to, and that wouldn't be a good thing. Now, when we look at an enzyme, it has an active site. The enzyme here is sucrase. It's the green. And you see it's got that little active site that's labeled. That's where the substrate is going to bind. And the substrate is defined as the chemical that is being metabolized by the enzyme. And you can see that the substrate sucrose will fit in that active site on sucrase. So the shape of the substrate has to match the shape of the enzyme's active site. When we talk about the induced fit model, that's where the enzyme active site changes shape after the substrate binds to it. And it kind of hugs the substrate so that it fits more snugly in the active site. And when it changes that shape just a little bit, you can see in number 2 down there at the bottom how it's changed shape. That makes that substrate split, and that releases the subunits. And then you see in number 3, the enzyme's gone back to its original shape. That's called the induced fit model. As I said, they're very specific. They only catalyze specific reactions. And it depends on the shape of the active site and the shape of the substrate. An enzyme, remember, just a protein. And so it's made of different sequences of amino acids. And the way that those sequences are set is by our genes. They only work under certain conditions. If the conditions are not correct, they will denature. They will lose their shape. They can't work anymore. Some things that will denature them would be the wrong temperature or the wrong pH. Now, warming it up a little bit might cause it to happen a little faster, but if it gets hot, it destroys it. If the pH changes, it destroys it. It will denature the enzymes. They will not work anymore.
In living organisms, most chemical reactions are catalyzed by proteins called enzymes. The three-dimensional structure of an enzyme is crucial to its function. If this structure is disrupted by exposure to high temperatures, for example, the enzyme stops working. The enzyme binds reactants in its active site, which is specially shaped to fit the reactants. The reactants on which enzymes act are called substrates. In this example, glucose is the substrate of the enzyme hexokinase. The enzyme and substrates are represented here in an artist's drawing. The substrates include the atom A and the molecule BC. The substrates enter the active site of the enzyme. By holding these atoms close together, the enzyme reduces the amount of energy the atoms need to enter the transition state. In the transition state, the bond between atoms B and C is broken, and a new bond between A and B is created. When the new bonds form, the products are released, and the enzyme molecule is ready to catalyze another reaction. Each enzyme is specific for only one reaction. For example, this enzyme could not catalyze a reaction between atom A and molecule BD. One model of enzyme-substrate interaction is called the induced fit model. It proposes that once the enzyme binds the substrate to the active site, the enzyme changes shape slightly to bind the substrate even more firmly. This places a strain on the existing bonds in the substrate, thereby lowering the activation energy needed to break the old bonds and generate new ones. This graph shows a hypothetical reaction between atom A and a molecule containing atoms B and C. The activation energy is the amount of energy needed to get the reactants to the transition state, in which bonds are broken and new bonds are formed. This transition state usually lasts for a fraction of a second. In this example, the products of the reaction are the molecule AB and the atom C. Note that in the reaction shown here, some of the energy stored in the reacting molecules is released. Often, activation energy can be a barrier that prevents a chemical reaction from occurring, except very slowly. The activation energy for many chemical reactions can be reduced with enzyme catalysts. Without catalysts, many of the chemical reactions of metabolism would occur too slowly to support life. Catalysts stabilize the reactants in the transition state, allowing old bonds to break and new bonds to form. Even though the activation energy is lowered by a catalyst, the amount of energy released by the reaction is unchanged.